Hello and welcome to this lesson for Higher Biology, which is looking at genetic technology. So specifically, we're considering which genetic technologies have been used to improve plant and animal varieties. And you need to be able to explain how DNA sequencing has been used in breeding, explain how recombinant DNA techniques have been used in breeding, and give examples of genes that have been inserted into crop plants. So plant and animal breeding has been occurring over hundreds of years, but genetic technologies have recently been used in order to speed up those processes or to make them more accurate. Both gene sequencing and recombinant DNA techniques are being used to do this. And gene sequencing essentially involves discovering the sequence of bases within an organism's genome. So you're going to remove one of the cells from the organism, extract the DNA, and sequence it and consider exactly what order of genetic bases you've got within that genome. What you're looking to do is generate information that will be used to identify the organisms that have the specific desirable gene sequences you're looking for. So if you're trying to breed crops with a specific resistance to disease, then you may breed 2 or 3 individuals together. And when you look at the offspring, you can't tell visually which ones have got that disease resistance. What you can do, however, is gene sequence their genomes and look for the specific gene conferring DNA resistance. You can then use those individuals in further breeding programs to produce offspring, also showing that desirable characteristic. As well as identifying genes in specific individuals to breed, genetic sequencing can also allow single genes for desirable characteristic to be transferred from species to species. Recombinant DNA technology can be used to insert those genes into the crop genome. Those individuals can then be bred as normal, transferring the useful characteristic to future generations. So remember, we're considering the fact that we can use gene sequencing to look for useful characteristics present in other species and insert them into our plants through the process of genetic transformation and recombinant DNA technology that we've looked at previously in Unit 2. Recombinant DNA technology in plant breeding includes insertion of the Bt toxin gene into plants for pest resistance. Bt toxin is naturally produced by a specific bacteria, and it's converted into its active form in the stomach of some insect species. That active form of the toxin destroys the guts of the insect and therefore kills it. Through genetic engineering, we can pass the gene for this toxin onto crop plants, where it will be produced in the roots, the shoots and the leaves. Any insect eating that plant will then be poisoned, but it's got no effect on plant growth. So that's been used in many really economically important crops like maize and like cotton. Similarly, the gene for resistance to glyphosate, which is a commonly used weed killer, has been inserted into crops such as maize and soy. And that lets farmers use glyphosate to control the weeds around them and reduce plant competition, but it doesn't harm the crop plants. So you should be able to explain that DNA sequencing has been used in breeding to identify organisms that have got the specific gene sequence you're looking for. That recombinant DNA techniques have been used in breeding to pass useful genes from species to species, inserting the genes from other species into crop plants in order to give them a particular advantage. That some of the genes that have been inserted into crop plants are the Bt toxin gene or the glyphosate resistance gene, and both of those will help increase the yield of the crop without particularly harming their growth.